fine. I'm very excited to be with you today. I'm Claudio Spero. I'm ophthalmologist by background, retina specialist, and discover the work in the pharma industry in gene therapy and then inherited retinal dystrophy. So I'm delighted to be here. I'm typically based in Switzerland, but I'm from Brazil, so I'm in my home country today. And got here this morning and then was online like an hour ago because I was just like going to get confused. And it's just a testimony of my willingness to be here with you and make sure that all the connection works well. Um, do you see well my slides, Bhavna?
Yes, I can see them really clearly.
Okay.
Thank you.
Sounds good. Okay, thank you very much. I'm leading the medical affairs team at AAVantgarde Bio. And before that, I had the pleasure to work at Novartis, rare disease, where I was leading the medical affairs team, working on the Vortigena Parfovec for the treatment with patients with IRD with ORP65 mutations. Here, we're talking about a platform that we can treat large genes. And then when we say large gene, is that the size of a gene which can be packed inside of a vector. In this example, it's a adeno-associated virus. So the example that I've just mentioned to you about Vortigen Nembarfovec, the medication is called Luxturna, that was treated for RP65, and it was small enough to get into the size of a AAV vector. When you talk about large genes, and it's an example of Stargardt disease and then Usher 1B, those genes, they're larger and cannot be packed in a single vector. So we have a platform to overcome this. AAVantgarde is a clinical stage biotech company. We are based out of London. And then our proprietary dual AAV platform is designed to deliver large genes. Large genes, those are the ones which does not fit in a single AAV vector. We'll get to that point a little bit later today. We have two clinical stage programs, one in Stargardt disease and one in retinitis pigmentosa caused by myo7A variances on Usher 1B. Those are in the clinical stages, and we do have a pipeline covering other IRD disease, which focus on large genes, but also in neuroscience and other areas as well. Bhavna asked me to cover a little bit about the eye, about the gene, about the inheritance of ABCA4, about signs and symptoms. So before going that, I would like to share just one image and some insights of the eye. It's a very unique organ, and then it has several functions, but if I would describe the most important function of the eye, is really to bring the light from the outside, go through all the tissues of the retina of the eye, and it's going to touch the very special tissue inside of the eye, of the back of the eye, which is the retina. And this tissue is very important because it has photoreceptors, cones and cells and rods. And these cells, they are specifically to transform the light impulse, which is coming from outside, into neuronal impulse. And in order to do that, there's a number of proteins and then enzymes which needs to be in place, one connected, functioning one after the other in order for the visual cycle to be completed. The ABCA4 protein, which is the root cause of the genetic disorder of Stargardt disease, is one of these very important and key proteins in which should be well functioning in the back of the eye at the level of the photoreceptors. Stargardt disease is a genetic condition. And I started by saying that Stargardt is an inherited autosomal recessive pattern. What does it mean? Inherited because typically in the majority of the cases would inherited those variances from our parents. And then recessive because we need to have both alleles with this which would cause Stargardt disease. Here in the bottom right, you have an example of the karyotype of the patient, an example of illustration of the 23 pairs of chromosomes that exist in all of our cells. We have two examples of each of those, with the exception of the X and Y chromosomes. They're different depending on the gender, but all the other 22 pairs, we have two examples. Here on the top right, you have an example in the left of the chromosome number 1, in where the ABCA4 gene is located. This is coming from my mother, and then the example of the chromosome 1 to the right is coming from the father. And then the little ray is just illustrating where the ABCA4 gene will be located. So in normal conditions, this gene is functioning well, and then the function of the ABCA4 gene is to produce the ABCA4 protein, which is extremely important for the life cycle of the patient, so the patient can see. In patients, we would have the condition of Stargardt disease. These variances on the ABCA4 would need to be affected both allele. So that's why we have an example of an error on the 2 allele of the chromosome 1, which means that there's a malfunctioning of the ABCA4 gene in both allele. Depending on the severity of these variances, it can be a slightly malfunction, a high malfunction, or the gene might not be even produced. But in all the cases, that would affect the vision of the patient, which will not be able to complete that visual cycle, which is just mentioned, which is important for the patient to see the light and images coming from outside. So, Stargardt disease is an inherited and rare condition. A prevalence, it will vary, but what we can see on the literature, it's between 1 every 8,000 to 10,000 people. And symptoms, they can start very early in age. Depending on the severity, depending on the type of variances that the patient would have, the beginning can happen in early childhood, but typically would get started around teenager, between the ages of 10 and 15. There are many signs and symptoms, but the only one that I would reference here more, it's the appearance of what some of the patient would describe as a black spot in the center of the vision. It's almost like you look through a window on during a foggy, and then you have the mist, and then you try to clean that, and it's not possible because this black spot, it's inside of the eye. And this spot will be in the center of the vision, and depending on the progression and then the time of the length of the disease, these black spots can increase in time. And then what is the consequence of this black pod is that when the patient would see, and then when the patient go to the ophthalmologist and do an assessment of best corrected visual acuity, which are those charts here in the bottom, it's when the physician would measure how how is the visual acuity of that patient. This patient would be able to see a smaller number of letters compared to previously in the system in the severity of the disease, or compared with patient without the pathology. So these black spots, it's in the center of the image, and then would make it harder for the patient to read, to recognize faces, and to see fine details. Another sign and symptom from Stargardt disease is what we call difficulty to adapt to dim light. So all of us, when we get inside of the theater or the cinema, we go from a very bright light into a room with a very dim light. And then there's a 10 seconds, 30 seconds of adaptation to dim light, which everyone would be a little bit troubled at that moment. But at some point, your eye will get used with that level of dim light, and then you can find a seat, you can find your colleagues that are around of you. One of the signs and symptoms from Stargardt disease is that this adaptation for the dim light conditions can be longer depending on the condition. One of the, we do have a number of visual function assessments, which is required to do a full assessment of the eye condition with Stargardt disease. And here I've just listed a number of the assessments that we would do in ophthalmology cabinet and to assess how your eye is performing. But I would like to start by clarifying that extremely important to get a genetic molecular diagnosis of the disease because that's where you, patient, the caregiver, and the ophthalmologist treating these patients would understand which are the variances. If these variances, they are in the two alleles that I described it, which means it's an inherited recessive condition. And then depending, the assessment of the genetic will tell the physician what if these variances are milder or more severe. So genetic diagnosis it's extremely important. And then the benefit of getting contact with patient associations like Stargardt Connected is getting guidance on how to assess and how to get this genetic test in the UK and elsewhere. Examples of visual function are best corrected visual acuity, here in the left. This is one of the basic assessments in ophthalmology. You ask the patient to sit at a established distance from the chart. You have a chart in white with dark black letters or signs, and then you ask the patient to read letter per letter. So that's what it says, best corrected vision. Best corrected because we allow the patients to wear glasses if they need glasses to improve their distance. So this is a measurement that all of the physicians would do in your clinic. In other assessments, we have the second one is low luminance visual acuity. It's the same of assessment of the visual acuity, but the ophthalmologist, the optometrist would put a filter in the front of the vision because we want to mimic the vision in dim light conditions. Typically, the vision in bright light conditions is higher or greater compared to the vision in lower luminance, but we want to assess how do they perform, how do they change over the life of this patient with Stargardt disease. And then we do have a number of images assessment. I've just put it here two examples. This is fundoscopy. This is a picture from inside of the eye. And then in very simple elements, here you have the optic nerve. And then out of it, you have the branches, you have a number of veins and arteries. And then all this red tissue is the retina that we can see. And then in the middle here, you have the disease of Stargardt, and we want to measure the size of this alteration and then compare throughout the life and the follow-up of the patient. Another way to assess these images is on fundus autofluorescence, is this image here. It's a different type of image and picture that we're taking. We're assessing different layers of the retina and how the healthiness of these different layers. And then we have a number of other assessments that we have here. We have assessments that ocular coherence tomography. It's an assessment that you go through. You do almost like a RMI from the retina and compare the different layers. And your physician will be the one most capable to explain you all this assessment. But once again, starting with a full genetic diagnosis of the disease, making sure that you understand which are the variances that you have in your case, and then trying to understand that those both alleles are attend of the ABCR4 gene is one of the points which is extremely important. Therapeutic approaches, we do not have any approved treatment for Stargardt disease for the moment. There's a number of type of treatments were being evaluated. including gene therapy, retinal implants, oral medication, stem cell treatments. AAVantgarde is investigating a gene therapy for the disease, and the aim of this can range from slowing or even stopping the progression of the disease. Patient association is extremely important. So when you're here today, get in contact with all the work that Bafana and her team has put together, having contact with patients, having talked to them, sharing experience, it provides support for not only the patients, but the families as well. It's important to share contact of specialists, information about disease, information about treatments which are under development. At some point, we really want to see treatments getting into the market in different formats. And then another guidance that it's coming from the patient association is on how to obtain genetic test. I would like to follow up on this discussion about very high level on what is the Vanguard. As I've mentioned, with our biotech on delivering large genes, and then we have a single dual AAV platform with two flavors. One in which we're going to connect the two pieces of the DNA in the level of the DNA, and the other one in the level of the cytoplasm. Bhavna, this is the moment that I'm going to stop sharing. I'm going to share again because I need to share sound.
Many patients live with inherited retinal diseases, yet vision impairment often goes unseen. The human eye is a marvel of biological engineering, where rods and cones convert light into electrical signals, enabling us to perceive the world in colour, depth and motion. IRDs remain an unmet medical need, as photoreceptors gradually degenerate, leading to progressive vision loss and underscoring the urgency for new therapies. Adeno-associated virus, AAV vectors, with their capacity to encapsulate a therapeutic DNA of 5 kilobase in length, offers hope. AAV vectors can be administered via subretinal injection, an intraocular procedure that delivers vectors into the virtual space between the retinal pigment epithelium and photoreceptors, creating a localized and reversible bleb. However, many IRDs stem from mutations in large genes that exceed AAV's 5 kilobase cargo limit. To overcome the 5 kilobase limitation, AAVantgarde has developed two innovative platforms, dual hybrid and intine protein transsplicing. Both approaches split the gene into two halves, packaging each half into separate AAV vectors. Inside the cell, they reassemble to produce a full-length protein. The dual hybrid strategy relies on a recombination event between both DNA molecules carried by the AAV vectors inside the nucleus of the same photoreceptor cell. Once the DNA is transcribed into mRNA, splicing removes the remaining sequence between the halves. The ribosome then reads the mRNA, producing a functional protein. Intine protein transsplicing follows a similar strategy. However, instead of recombining DNA, each AAV expresses an mRNA molecule that is translated into different protein fragments. Thanks to the intine sequences, the two protein fragments self-assemble, forming a complete and functional protein, while the intine fragment is excised. With these advances, there is potential to restore photoreceptor function and vision. AAVantgarde is paving the way for transformative gene therapies, bringing hope to those affected by IRDs.
And Bhavna, I'm sharing again my screen because I will go back to sound. Okay, from a clinical development perspective, AAVantgarde has two studies on Stargardt disease. Stella, it's a natural history study in which we are following patients without treatment, trying to understand what is the disease progression of these patients before any type of treatment. We are doing this study in these different geographics in the UK, United States, in region Europe, and then we are just about to start our interventional study. So Celeste is our phase 1, phase 2, phase 3 interventional study in which once the patients they have been treated at Stella, they would move into interventional piece. This is a study that we would start with adults and then expand to pediatric population and adult population. And then we would investigate our treatment for Stargardt disease, which is AAVB 039. I'll stop here for the moment. I'm delighted to be here today, and Bafana asked me to come back early December for another webinar where we're going to be able to go in more in depth into the discussion about our clinical development program, and I will be delighted to go in depth a little bit more on the mechanism of action of the disease. Bhavna, I would like to thank you very much for inviting me today. If you have any additional question, I'll be delighted to take here on the Q&A, or if you'd like to contact AAVantgarde, you have here our email box. Please don't hesitate to contact us. Thank you very much.
Thank you so much.